friends. Today I'm going to be discussing genetic scientists. You may know of Gregor Mendel, Thomas Hunt Morgan, James Watson, Francis Crick, Erwin Chargaff, and a few other familiar names who contributed to genetics as we know it today. But my main goal is to share the discoveries and contributions of a few scientists whose names you may not be familiar with. First up, we have Jean Brachet, a biochemist from Belgium. In the early 1930s, Brachet and his co-worker, Swedish geneticist Torbjorn Caspersson, were able to detect the presence of RNA in living cells. They also saw and agreed that more protein synthesis in the cell correlated to its increased levels of RNA. Caspersson proposed that the nucleus was a key site of protein synthesis in the cell, but Brachet disagreed. He removed the nucleus from Amoeba proteus cells, and protein synthesis still continued for days inside the cytoplasm filled with RNA. Thus, he was able to prove that RNA played a very active and essential role in protein synthesis. This challenged the previous idea that the nucleus filled with DNA directly synthesized proteins, which led scientists closer to the central dogma of biology, where, quote, DNA makes RNA makes proteins, in 1940. Next up, we have Jean Brachet, an Indonesian-born American geneticist, who embarked on his scientific work after spending 3 years in a World War II Japanese internment camp. He discovered that there were 46 chromosomes in the human body in 1955, not 48 chromosomes, a belief that had existed for 50 years prior to that. Dr. Brachet was studying embryonic lung tissue and had been trying to redefine the technique of separating chromosomes onto a glass slide, when he counted the chromosomes and realized that there were 46, not 48 chromosomes. Scientists had already understood chromosome function beforehand, but Dr. Brachet's finding helped others discover in 1959 that people affected by Down syndrome had an extra chromosome in their cells. He was even rewarded by JFK with an Outstanding Achievement Award in 1962. Finally, we have Herbert Boyer, who in 1966 became assistant professor of microbiology at the University of California at San Francisco, the capital of hippies and counterculture. He became interested in the types of restriction enzymes that can be isolated from an E. coli cell. He collaborated with Stanley Cohen in the early 70s, and the two were able to create the first piece of recombinant DNA, consisting of a plasmid holding the desired DNA fragment. Using the plasmid to hold the DNA, they were also the first to figure out how to clone the recombinant DNA. In 1976, Boyer founded Genentech Incorporated. Using this new DNA recombinant technology, he and his business were able to produce masses of commercial medicines such as synthetic insulin. This was made by constructing a plasmid that coded for insulin, and it became their first genetically engineered product, followed by many others. Genentech is now one of the biggest biotech companies in the world.